Alright, hello everybody. Welcome back. We are on lecture 6 right now, going over DNA topology. Um, yeah, just a reminder, workshop on Tuesday the 20th. I've recorded a lot of videos in one day. This is the last one that I'm recording for today. So, yeah, let's go ahead and get into this. So in lecture 5, we talked about different nucleases. I'm not going to go over them again, but you've got exo and endonucleases. Restriction enzymes are a subclass of endonuclease. There's a whole bunch of them, yada yada yada. There's also DNA ligases. They connect DNA together. They use ATP or NAD+. And they're not really specific, and they use double-stranded DNA as a substrate. And yeah, they use a lysine nucleophilic attack mechanism where they basically attack, form a covalent bond with the DNA, and then with the ATP, they form a covalent bond with A and P, and then from there the DNA attacks, and then they basically use that double phosphate group that's connected to the enzyme to initiate a nucleophilic attack and like a transesterification reaction kind of thing. If none of that made sense to you, go back and watch my Lecture 5 video and skip to the end where I talk about that reaction. But yeah, let's go ahead and move on to DNA topology. This lecture is pretty confusing, I'm not going to lie. Even I'm a little bit shaky on it. But, understanding DNA supercoiling at cellular functions, how does it work? Linking number and all that stuff, and then comparing types of topoisomerases. This is a bit of a weird section, so... First, we have to ask ourselves, what does DNA naturally look like at a large scale? And the answer to that is it looks like a mess. It's very compact, generally speaking, especially when it's in the chromosome form. Like, chromosome is the most compact version of DNA you'll ever get. But, on top of, DNA, having this, a helix structure, right? That's not a good example, but on top of DNA having this, helix structure like this, the DNA can also fold on itself. So imagine that you can kind of see it here, but you see how there's twists inside of this DNA, right? But there's also twists on top of those twists, and that is supercoiling. Essentially what is happening is we have the double-stranded DNA kind of doing its thing, but then it also is able to form macrostructures on top of that twist that's already happening within the DNA. And so what you get is these massive messes that look like this. I don't know if I can outline this for you, but it's kind of hard to see. But it's basically twisting on top of each other, and it's doing all sorts of weird nonsense. And that's where DNA is kind of weird, and it can do a whole bunch of stuff. So it's DNA supercoils are very flexible. They can change shape really easily, and the topological properties of DNA don't change with the DNA shape, but the speed at which it flows through a gel, for example, like a DNA gel, might change, depending on how compact the DNA becomes as a result of that. So, like I said before, supercoiling is required to make DNA compact. So you can see, right here on the left side, this is a very loose piece of DNA, but as we start to twist it on itself, you can see it becomes more and more compact, and now it's just like a straight line. It looks like a straight line. And so when we say that all DNA is naturally negatively supercoiled, I believe that means that it's supercoiled to the left. And so the easiest way to actually to do this and to test this is to take a string or a cord or something, and then start twisting it. And you'll notice that with time, as you twist it, it starts to make these sort of weird kind of bends that aren't normal. I'm actually using my own lanyard here as an example. You obviously can't see it, but if you were to do this with me, you'd be able to see that as you continue to twist, the twisting would start to contort the actual lanyard or whatever you're using, and then those contortions that cause little loops are actually what we call supercoiling. And so that's what actually makes it tighter. So, supercoiling is very important for packing. Like I said before, I'm not going to go into more detail about that, but you can imagine, right, that it's a lot easier to stack this than it is to stack this, right? It's just more compact. So, there you go. I won't bore you with this, but yeah. Supercoils for packing, I already talked about this, right? I mean, there's also like histones as well. Histones are important. So like when you do the supercoiling, you can add histones in there that helps with um, that helps the supercoiling kind of stay stuck in that way. So like you can supercoil, right? And when you supercoil, if you add these histone proteins in, they actually help keep the supercoil format very stable. But see, here's an example of what this looks like. If you take a string, right, and you continue to twist it, it's relaxed in this form here, but when you twist it a lot, it overwinds and you get these sort of like weird artifacts in the actual supercoiling, and these are what we call supercoils right here, and you know that doing those supercoils actually lead to slight relaxations that allow us to pull the DNA apart, and that's actually how DNA replication forks work, in a way. So here's an example, sort of how RNA polymerase works. Essentially, what's happening here is that while this is RNA polymerase is traveling this way and splitting the DNA, this area over here is like, you can imagine that if you were to pull on the two ends here, eventually it would like tighten up and it would become hard to pull. That's what's happening here. You're basically pushing this, and this event becomes overwound, very positively supercoiled, and then if this becomes positively supercoiled, then this has to be negatively supercoiled. Basically, it's like what happens to one must have like an opposite reaction from the other one, essentially, if you want to think of it like that. So here's like an example. So like this is a bit of a weird example, actually, now that I think about it, but I'm try and work with this. So let's see what's going on here. Yeah, so I don't know why they've decreased the number of turns, but yeah, that's fun. I'm actually going to move past this. I feel like this image is more confusing than it needs to be. It's compaction and strand separation. I already talked about this a little bit, right? Sort of. Basically, when you negatively supercoil, you basically loosen your DNA a little bit, and so it allows for you to pull them apart. So I won't go further than that. I feel like if I do that more, it's just gonna become really confusing, and I don't wanna confuse you guys. Okay, this is what actually matters. This is what you'll probably actually be tested on: linking number, twist, and writhe. So there's two types of, I guess, supercoiling, if you will. Twists are what we normally see in DNA, right? Like the whole idea where it like twists on itself, right? This whole structure where it does this kind of thing. That is twist, right? These sections here, they're twists. Writhe is when it does this. So like you can imagine that each of these, right, looks like this. Like there's like little, it looks like this in each one of these small sections, right? Writhe is when that basically overlaps itself and like twists again, if you will. And what you can do is, if you're given a number of base pairs, what you can do is you can take that number of base pairs, divide it by 10.5 base pairs, and that'll give you the actual number of twists within your DNA, assuming that it's fully circular or like, and there isn't any like supercoiling involved in it. So that's kind of how you calculate your linking number if you don't have if they don't give it to you. So it says that writhe and twist can be interconvertible, and this is true, right? So essentially what you can do here is, I don't really know why they're doing it like this, but there's one thing about these lectures that kind of annoy me, is the structure of them. But I believe this is supposed to be, is it 19? And then minus 2? All in all this to say that, you know, writhe and twist are interchangeable. So they're not the same thing, but they are interchangeable. So the way that you calculate, and the way you look at this is the linking number is going to be the combination of the twist and the writhe. It's an addition of the two. It is a constant number for a closed circle of DNA, and it does not change. The only time it changes is when you cut the DNA. When you cut the DNA, then it kind of, all that goes out the window, and then you end up with an undefined system like here. See, like right here, see how it's nicked now? Because it's nicked, right? It's no longer a constant, and because it's not a constant anymore, we have to say we have no idea, so it's undefined. So, the only one I would really worry about is, I wouldn't really worry about these three down here. I would worry about this one. This is the most important one that I would worry about. You can keep these on your note card if you'd like, but I was able to do well in the class just by looking at this equation here. Realistically, on an exam, they're only really going to ask 1 or 2 questions about linking number. But yeah, so you can see here, if we split this, right, and we look at these 4 here, you'll notice that in this one, we've got -3 twist, and so and 0 writhe. So that means our linking number is just going to be -3. And so we can also get this by removing one of the twists and adding a negative writhe. And we can also keep going. We can add another negative writhe. We can remove another twist. And then we can add another twist and then remove it. We can remove another twist and then we can add another writhe. So like a negative writhe. So you can basically, these structures are very different from each other, very obviously, but their linking numbers are the same. The other time I want to point this out, when linking number is undefined is when it's linear. Because linear, right, there is actually an end, and so because there is an end, it can unravel freely. That's kind of the big issue with if the DNA can unravel freely, it's going to be linear. So this is another thing, right? So you can have DNA segments that are constrained. So if they're constrained in a closed circle, right, then it's probably going to be a constant linking number and not undefined, right? If it's constrained. That's kind of the main thing they're trying to get through with this. I think there's a lot of extra information which I feel like is not that important. What you should understand from this is that when DNA is closed, and when it's circular or when it's like closed off, the linking number tends to stay constant. And when you snip the DNA or when it's open, then the linking number is undefined until it's closed again. This is also not very important. I think you should just keep this in mind. So if you have right-handed supercoils, they're called plectonemic. And then if you have left-handed turns, it's going to be called solenoidal. These are like, yeah, so to put this simply, this is just how, so bacteria, for example, they use this plectonemic kind of right-handed coiling, but in eukaryotes, we often use this solenoidal kind of supercoiling instead. We use this left-handed supercoiling because it's more compacted and it's more easily stabilized by binding proteins. Again, how important is this? I wouldn't say this is super important. I would just try and remember these words. Remember that solenoidal is left-handed, and plectonemic is right-handed. Okay, this is where we get into some of the enzymes. So there are some enzymes that can change DNA topology and the writhe and twist of these of DNA. And these are topoisomerases. And topoisomerases are actually very important because they are actually used in DNA replication as well as some other things as well. So we'll first start with topoisomerase 1. And topoisomerase type 1, what it does is that it relieves tension and torsional stress. And what happens is it doesn't really just hydrolyze it. It's not simply hydrolyzed because... Yeah, so the important thing to recognize is this actually doesn't use ATP, which is why this is important. It doesn't do like a hydrolysis reaction, because if it did a hydrolysis reaction, it would... If it did a hydrolysis reaction, what would end up happening is you would have to use ATP, and you would actually end up using energy for this. But instead what we do is we actually do this thing where we take one of the DNA strands that are in the active site, and then what we do is we attach a tyrosine onto the end of one of these strands, and we essentially break the strand. And that mechanism is here, right? They show it here. Essentially, what's going on is this tyrosine attacks this phosphate, and then we're left with one side of the strand up here and another side of the strand up here, down there. These are two single strands, by the way. They're two complementary strands. So one complementary strand, and then the other complementary strand. And so when they do that, there's a conformational change that occurs. And so we end up with this situation where there's a cut. And now what happens is that the uncut DNA, this little finger kind of thing here, uses momentum to kind of push the uncut DNA through it. And then from there, that base that was the cut that was originally there is now fixed up again. Like the O from this attacks the phosphate and it kicks out the tyrosine and resets the whole thing. And so basically you've basically just relieved a twist by doing that. Yeah, so you've essentially what's it called? You've released a twist by doing that, because you've essentially cut that DNA, and when you've cut that, you pull the strand through. You pull this strand through, and then once you've pulled it through, there's no longer that twist that was there before. I hope that makes sense. It's kind of hard to explain, but maybe if I do it like this. So imagine that you cut this section right here, and then you bring this strand through like that. What you'd end up with is this. Right? Do you see how we have... Actually, I messed it up. Wait. You'd basically end up with this. A straight line, if you will. So we've gotten rid of that twist, if you will, right? We've gotten rid of this singular twist in this area. So that's how I've always thought of it, and it's helped me a lot. So if it works for you, go ahead and think about that. Think about it like that. But if something else works better for you, feel free to just use a different method. The other one we have is topoisomerase 2. And topoisomerase 2 is a bit different. And it relieves supercoiling actually by removing two twists. And the way that it does this is it actually takes full double-stranded sections of DNA, uses ATP to kind of hydrolyze, and then it pulls one DNA strand through the other. So you can see here, this green and this purple. You can see here that the green is kind of there. We've put some ATP in there, and that ATP is used to snap the green here in two. And then once it's been snapped in two, this purple is pulled through. You can see that right there, right? See the purple is pulled through there. And then from there, we have to use... and then from there, it's pulled through, and then the green is kind of pulled back together again. The other version of topoisomerase 2 is this version, where you have your pink, you have your green. The green is snapped in two. Use ATP, pull it through, and then reanneal back together. So the ATP is basically used to break the DNA and also reanneal it, if you will. So the main thing you should know is that these mechanisms, these topoisomerase 2s, they relieve two twists, essentially. They relieve a supercoil. They relieve a supercoiling by removing two twists, and they use ATP in the process. This is very different from topoisomerase 1, or type 1, does not use ATP. Topoisomerase type 1 does not use ATP. No ATP. It's the main thing you should remember. These both, any topoisomerase 2 is 2 ATP. Always. DNA gyrase. DNA gyrase is one of those ones where I have a hard time trying to explain. It's basically a unique type of topoisomerase 2. And it's special because it's specifically a bacterial type of topo 2. And similarly, it also uses ATP, if I'm not mistaken. Yeah, it uses ATP. And if I'm not mistaken, I think this one is weird because it actually introduces supercoiling rather than gets rid of it. Because most of these up here, they actually get rid of supercoiling. They actually get rid of negative supercoiling. But gyrase is interesting because it actually introduces it. And so what you get is very similar mechanism where you have our linking number of -4 in this case. But what's going to happen is this multi-subunit enzyme is going to basically hold on to this DNA. It's then going to use ATP to cut one of the double helixes, and then pull the and then pull these two through it. Pull these two strands through it. And then that actually ends up introducing more supercoiling in comparison to how it was before. It's a really interesting mechanism. But yeah, I would say you don't really have to know the mechanism for how it works. What you should know is that gyrase is a unique type of topo 2 that introduces -2 supercoiling rather than unlike most topoisomerases that remove supercoiling. This one actually introduces it instead. But yeah, the mechanism is a bit weird. But yeah, so you can see here, like it explains it here, but like firstly the enzyme binds to the DNA segment in question, so it'll bind to this tan region. It's then wrapped around the complex in the end gate. So it wraps around this section here, and it stabilizes that, and then the bound light red is cleaved across both strands. and then the purple DNA passes through the break and exits the enzyme through the C gate. So it basically, like, you cut the tan, you pull the purple through, and then because of that, you have this weird thing where if you pull it through and you re-anneal it, it does this weird thing where it like coils back on itself. But again, the mechanism is not that important. I would say that just knowing that it uses 2 ATP and the fact that it introduces supercoil. I'm just going to say SC. Introduces supercoil. This is unlike the other ones that usually remove supercoiling. And what I want to make very clear about this slide specifically, is how supercoiling affects speed of DNA travel. So you can imagine that when you run a gel, the smaller things actually go through the gel quicker. Now, in this case, say that all of your DNA is the same size. What's going to differentiate what goes through quicker is actually going to be whether or not it's compact and supercoiled or not. So, see something like this, right? It's not very supercoiled. That's actually going to take longer to pass through than something like this, compared to this, compared to this, compared to this, all the way down to this, which is the most supercoiled. So that will actually travel the farthest. And so all this to say that the thing that travels the farthest, if you have DNAs that are all the same length and you see multiple bands, it's probably because some of them are supercoiled and others are not. So that's one of the main things you should keep in mind. So it may not be that the DNA actually represents different sizes, but it could be that the DNA represents which of these DNA fragments or DNA molecules are more supercoiled than the others. And the answer is that the one that's traveled the farthest, assuming that all the sizes of the DNA is the same, is that the one that travels the farthest is the most supercoiled. And this is a nice little slide just to talk about topoisomerases as drug targets. So topoisomerases are actually very important for DNA replication. And so the thought process behind these drugs is that by blocking topoisomerases, DNA replication can't occur. And so if DNA replication can't occur, then the cell count divide, and that's inherently what cancer is, right? Uncontrolled cell division. And so no cell division means no cancer. But yeah, other than that, I hope this video was helpful. I know that this stuff is a little bit weird. I don't think I did a great job explaining it. So if you have questions, feel free to ask in the comments or come to the IC. Oops, just bumped my mic. Feel free to come to the IC, ask me questions, or ask anybody else at the IC, and they can also probably explain it to you. But yeah, I hope the video was at least somewhat helpful and you learned something. But yeah, happy studying, guys, and see you in the next one.